Today I want to talk about two topics that I feel are really game changers when it comes to DOE. These are two topics that I wish I had access to when I was leading R&D efforts, chemical and product and process development, leading R&D in those areas. Um, and the first of these is going to be, we're going to talk about functional data and how we can use that with DOE. So where we'll use a curve as our response. We're not just going to use a scalar value like a yield or viscosity or something like that. We're going to optimize a curve response. And the other is self-validating ensemble models. So self-validating ensemble models, or SVEM, is not something specific for DOE. It's really a nice method for validating models based on small data sets. And it's just that those data sets are exactly what we're generating when we run a DOE. So it's really a nice modeling tool to help you get more out of modeling your DOE and giving you a statistical method for validating a model, not just your confirmation trials. Um, all right, so just like the other sessions, we're only going to scratch the surface today on what you can do in these two topics. And I'm going to go through them pretty quickly. We will have videos so you can go back through them and watch, but I really want to go through like a simulated workflow, how you might use these. And so we're going to go through it pretty quickly. But if you do have questions, we've got the Q&A at the end. And if you do wonder how you might be able to apply either of these methods to the experiments that you're running, you can reach out to me or Matt and we can schedule some time to talk. So let's go ahead and jump into functional data analysis. So first of all, what is functional data? It's probably a term that you're not familiar with. I know I wasn't when I learned about it, but I can probably guarantee you that everybody's using functional data. So if you're dealing with time series data, maybe looking at some response over time or a concentration of some value over time, this is functional data. So it's just a XY function. And it differs from scalar data that we're used to because we're not just looking at a point, maybe the concentration at 10 minutes or the slope of a line, but we're looking at this entire curve. This can also apply to non-time series data like stress strain curves or spectral data. So this is a type of functional data that we can use when we're doing this type of analysis, or particle size distributions or HPLC traces, things like this, chromatograms. So any type of data that you can plot on an X and Y axis is really functional data. And you're probably using it today and you might be optimizing it, but you can't necessarily optimize this curve when you're running a DOE using standard methods. And so let's take a look at this particle size distribution and see how, if we wanted to optimize some particle size distribution using traditional DOE, how would we do that? Well, we'd take a look at this curve and we'd see we've got an x-axis which has our size, so the micron, and then the percent per micron is our y. And we might say, okay, well, let's target an average particle size. So we might look at this, the peak, the midpoint of this curve and say we want to get this as close as we can to 90. But that doesn't really tell us anything about the width of this peak. Maybe we need a narrow one. So then maybe we'd also optimize another parameter, so some width at a certain height, like the width at 50% height, or we'd have to look at peak area quantiles, or maybe even just qualitative assessments. Because how would we mathematically or describe this little shoulder? So maybe in our analysis, we'd try to have no shoulder or little shoulder or something, some qualitative assessment. And so we lose a lot of data when we parameterize this curve. And so we're going to show how using Functional Data Explorer, we can actually optimize the whole shape of this curve. We don't lose any of the data, we use it all. And it's a really cool technique that we can now do in JMP. And so this example that I'm going to talk through is a milling example. So we're going to optimize that particle size distribution, and milling is really just taking large particles with a variety of shapes and sizes and making them to smaller particles with more uniform features. And we're going to use this simulated ball mill. This is some simulated data that we created, but we've taken some factors that are common, like agitation speed and flow rate and things like that, and we've created the factors for our design. And in this case, though, what's going to be our response? We could have that peak height or the width of the peak, but in our our DOE, we're going to actually use that curve as our response. Not only that, we're going to have a hypothetical curve. Maybe we've never been able to make a particle size distribution as uniform as this, but we can draw that and just use that as our target. And then when we optimize our parameters, we're going to optimize towards this target, which is a symmetric distribution that's narrow. And so let's go right into JMP and see how we would do this. So the goal of our functional data analysis is really to get, just like we do in any DOE, is to get to a profiler. So before I show how we get to here, I just want to show you where we're headed. And in this case, just like in all the other DOEs, whether it's a definitive screening design or a simple factorial design or a custom DOE, we're going to get a profiler that has all of our factors down here. So those things like agitation speed and flow rate and the media types, so categorical factors. But instead of a value that we're optimizing or a value that we're getting out, we're actually going to have that curve in our profiler. And as I move this agitation speed, I'm going to see how that affects this curve. And so if I go in lower agitation speed, we can see how that curve is changing. And if I go to the higher agitation speed, I can see it getting more narrow. Or if I look at this media loading, if I go lower values there, you can see the curve is flattening out. And if I go to higher values, we're getting more narrow. And because I've got a target curve, I can still use that red triangle optimization, maximize desirability, and JMP is going to try to find that the set of conditions in my factors that are going to get to that narrow symmetric distribution, particle size distribution curve. So this is where we're going, and it's really powerful. And if you have data, we've got examples where we are working with spectral data, with particle size distribution, viscosity curves, all sorts of reaction kinetic curves. So any type of two-dimensional data can be that target curve that you can optimize and relate that to the factors in your design. So let's see how we set this up. It does take a little bit more of a setup, but it's really powerful. So let's go through there, and I'm just going to hide those for now. And so we've got our particle size distribution, the factors for this design in a factor table that's all set up, and the type of design doesn't matter. You can do a custom design, you can do classical design. In this case, I chose a definitive screening design, that like something we talked about a couple sessions ago, I think. And so what we'll do is we'll just go to the red triangle and load those factors. And so now we've got all those factors in here. Click continue. And we can go with the default with maybe in this example, we can afford some extra runs. So we'll just make that design. We'll double check the design to make sure it's got what we need, and then we can make that table. And when we make that table, just by default, we're going to have this Y column in here. So it's hard to put that curve in this Y column. So we actually don't need it. So we're just going to delete it. Because we're going to pull in that two-dimensional XY data from another table. But this would still be our run sheet. So we'd run number one, and then in another data table, we're recording that final XY curve, and so on and so forth. And so what I want to do is just minimize this and show what our two tables would look like at the end. So our first table is our design table. So this is very similar to the table that I just worked with, but in this case, I've got a run number as a column, and that's going to help me link up to my other data. But aside from that, my DOE is simply just the factors. So just the factor settings and that run number. All of my curve data is in another data table, and I can take a look at the column headers here to see I've got 19 curves in this data table. They're all stacked up. We can see we've got nearly 3,000 rows. And then we've got the size, so that the x-axis and the y-axis here. And then we've got a curve type. And most of our curves are those experimental ones, but we do have a target in here. So we can take a look at the results from our design just in Graph Builder. And this purple curve is our target. So this is the one that we said we wanted to match. And and so we've got all of these blue curves are our experimental curves. And so you can see in this design, we've got a pretty wide range of response. We've got some that are kind of wide and flat. We've got some that are, pretty distinctly bimodal. We've got some that do have that shoulder on the edge, so it looks like, we've got a pretty wide range of results here, and we want JMP to help us figure out, what combination of settings are going to get to this sort of curve. So the way that we do that is we need to link these two data tables together, and you can do that physically, do an actual table join. I like to use something called a virtual table join. So you can see this little blue key here. That means it's looking for a table to link to, and it's going to link to this table that has that yellow key. And we can talk about how to create this virtual join in the Q&A if anybody wants to do that. But it helps me keep it nice and clean. So I've got my DOE data in one table and my results in another. But you can see that I've referenced in this data table, I actually have all the data from my DOE table in here. So what I can do now is I can launch the Functional Data Explorer by going to Analyze, Specialized Modeling, and we're going to come down here to Functional Data Explorer. So this is how we're going to analyze the DOE. There's a couple different data formats that you can use. Ours is in this stacked format, so we just have two columns that have our XY data, and then we've got an identifier that's going to tell us which is which. So we're going to put our size as our X, micron as our Y, and then our identifier is the run number. So we'll put that in there. And then all of those DOE factors now, I'm going to load in as supplementary information. And when I'm done with that, I can just click OK. And so now we've launched the Functional Data Explorer. And so we haven't done any analysis yet. It's really just loaded in all of our values. And there's a lot of things that we can do to help clean up this data if we have messy curves or align it. But and this data is pretty clean, so we don't need to do that. But what we do need to do is identify where that target function is, because we don't want to create a model that includes that target function. And so I've highlighted that one in green here. And so to get rid of that out of the system, I'm just going to go to target function, and I'm going to load a target function, and it just happens to be number 0, and I click OK. So now I've pulled that out of the system that I'm going to analyze, and by designating it a target, it's also going to be what I use in the profiler to optimize against. So right here, the only thing we just have all of our data, and then we've got our mean. So if we just take the mean value of all those curves, we get this, and then we've got a standard deviation curve. We haven't done any analysis yet. And this is where I'm going to skip through a few steps and just show you how to get to that end result. So what we're going to do is we're going to go create a model. And so I go to red triangle and models. And there's a number of different models. And if you're not sure which one is the best, you might need to create a few different models. I know that this wavelet model is a good way to represent our data. And so I'm just going to select that one. And you get a little warning that your data has to be organized a little differently in order to build the wavelet model, but JMP does that automatically. And so now we've got our list of wavelet models that we could use, and then JMP has highlighted what it's found to be the best one, the best fit to our data. So we're just going to go with the default. And if you're familiar with principal component analysis, then some of these plots might be look a little similar. But essentially what we're doing is JMP has looked at our data and said, okay, let me take this mean function that we start with, and then I'm going to create all these other different shapes. And we're just going to add or subtract various weights of these shapes to that mean function and try to reproduce every curve that you have in your data set. And so with only one curve, if we only use this one shape, we would explain about 60% of the variance in our mean function or across our data set. If we use two shapes, we're going to explain 87, and up until we can explain about 99% of the variation across all of our curves if we use 7. Now we could adjust this, and maybe we only need to use 5 functions or 4 functions even, but we'll just keep them all here at the default for now. But you can see as you get more and more functions, you're really starting to fit more and more noise. And so it just depends on, you know, how simple you want this model to be. We'll go with all of these. And what we can do now is we get something called a score plot. And I'm going to just show what this helps us visualize. And this is where you can just go and check your model. So we have a profiler here now that has our predicted curve and then the portion of each of those shape functions down here. And so as I move one of those shape functions, so here's shape function 1 and shape function 1. So if I move that to a lower value, you can see how the curve is changing. And if I move it to a higher value, you can see how the curve is changing. And we can just do spot check some of our curves. So if I look at curve 7, for instance, I can move that over here, you know, side by side. So this is the actual data, and this is my, oops, and this is my predicted data. And so if I click on this, it's going to put in all of the shape functions that are applied to number 7, and I can just visually check and see, yeah, those are pretty close. And I can do the same with, you know, function 6 and click on it. And so 6 is being predicted pretty well, is right there, and, you know, an 8, or whichever ones we might want to do. So we can see that we're able to represent a lot of these different features, whether it's just a shoulder or this bimodal nature of those peaks. So if we're satisfied with this model, we can now relate it to our DOE factors, because you can see we're not related to any of these DOE factors yet. We've only related this shape to those functional principal components. So what we're going to do is go to the red triangle, functional DOE analysis, and that's running something called generalized regression that relates those functional principal components, those shape functions, now are related to our factors. And so through that extra step, now we get that profiler that has the agitation speed here. And I can now see if I move that agitation speed, I can relate what the predicted shape is going to look like for that particle size distribution. And so that's how, that would be the normal workflow for going for this two-dimensional functional data. It's really powerful. You use all your data, and I really don't know any other modeling tools that can get you this type of analysis. So this is not going to be applicable to every DOE, but when your shape of your curve or your results are telling you something about your system, then this is a great way to start learning more about the system. Use all of the data and not just the parameters that you might extract from that data. All right, so I know that was really fast. We can always talk about this more, but I really also want to get into the second topic today, which is self-validating ensemble models. So just going to clean all this up and go on to SVM. So we're switching gears. SVEM is really not related to DOE specifically, but it's a new method for validating statistical models. And typically we validate statistical models using, you know, a couple different methods like a holdback method or cross-validation method, but these methods require us to hold back observations in our data set. In a DOE, every observation is critical. It was used to create the design, and taking those out can break the design, or you might not have enough data to generate a model. So you can't really use these types of statistical validation methods on a DOE. And that's where SVEM can really help. SVEM is really a way to give you a framework to do some statistical validation of your model. You know, as experimentalists, we're probably always going to validate our model with an experiment. You're going to run a confirmation trial, or you're going to do an augment design, or something to that effect to make sure your model is valid. SVEM gives you some ability to now statistically validate that model. There's some, I would say, mathematical wizardry going on in this method, but it really is going on in the background. You don't have to do a lot of this. JMP is doing this in the background, and it really lets you use each of the observations in your DOE to serve as both a portion of it in the training set and the validation set. And so once again, I want to talk about just the goal. It's like, what are we trying to do? Ultimately, we're trying to get to a model faster. We're trying to uncover, those active model effects, and we're trying to develop more accurate models. And I'm going to go back to an experiment that we talked about in the definitive screening design, because as I was trying to figure out a good data set to use for this presentation today, when I started analyzing this, I got an interesting result, and I thought I'd share it. And so when we did our definitive screening design and our stepwise regression, we came up with pretty similar models, with this methanol squared term. And if you weren't with us when we talked about this, we'll take a step back and talk about what this experiment was doing. But we found, some good agreement here on the curvature in this system. And, we found out what effects were active and what interactions we might have, and which is all the purpose of that definitive screening design, which is to find those active effects. When I ran SVEM on it, I found different active effects. So instead of having a methanol squared term, it's just got a methanol active main effect, but then it's explaining the curvature in the system with this propanol squared term, which I thought was pretty cool, because it's really highlighting why SVEM is pretty powerful. It's giving you a broader picture. It's another tool that you can use to find active effects. It's going to give you insights into that system that you might not find until you start going down the pathway of validating through experiments or additional trials, things like that. So I want to walk through how I create these models and talk a little bit about what's going on in the background, how we can actually use the same observation for both the training and validation set. Once again, we're going to go through this pretty quick, but I really just want to get this out there and just make you aware that this tool is something that you might find helpful in the models that you're creating. So in this case, we're going back to our buddy in the lab who's doing an extraction. And as part of this extraction process, he's looking at this extraction step where he's got different solvents, pH, and time as the factors. And so what we've got is the results of this trial. And so this is the same design that we looked at when we analyzed the definitive screening design. So we have all of our factors here and the yield from each of those trials. So what happens in SVEM is, so when we run a model of this, all of this is being used in the training set. So 100%, it's all, there's no validation when we do, a standard least squares type model. When we do SVEM, what it's really doing in the background is essentially duplicating your model and using one portion of it as that training set, and then it duplicates the entire model and calls it the validation set. Now, that's not going to be too helpful if all you used was your yield, because all the values are identical. So what it does is it breaks this up into different weights. And so it's saying, okay, for this observation, for this first observation, I want to put a larger portion of it into the training set and a smaller portion in the validation set. And we can visualize that just by looking at Graph Builder. And we can look at each of these observations. So observation 1 has a larger portion in the training and a smaller in the validation. And then we can look at number 2 over here and see how it goes. And you can see that typically these are adjusted so that there's larger proportion in one than the other. So if it's got little effect in the training, it's got a larger proportion in the validation, and then vice versa. So larger portion in the training set and a smaller in the validation set. And so we can use this this weighting to generate a model, and I'm going to use a Jump Pro modeling platform called Generalized Regression, and specifically a lasso model. And in this case, we can generate a model from this that's been validated, so we get a predicted versus actual for both a training set and our validation set. But this model, in this case, only has those 4 terms. And maybe we didn't find all the active effects with this one, because the different weightings here are going to give you different active effects, different parameter estimates. So what we can do is run this model, and then maybe we'd save it to the data table, and then we'd change our weights. So I'd recalculate my weights, and when I recalculate the weights, I'm going to get a new model. And this new model might have a different number of parameters in it. It's going to have, and those model effects are going to have different parameter estimates. And then I might do that again, save this to the data table, do it again, have another model, and then again with another model. And if we do this enough times, we can average all those together and find out what model terms are showing up the most in each of those models, and what's the average parameter estimate, what's the frequency that it's showing up in the model. And that's what the SVEM platform does in JMP. So in the SVEM platform, we don't have to do this manually. We can just go to our extraction results. We have our yield, and then I'm going to go to Analyze, Fit Model. We're going to load our model factors in with our response, and then I'm going to go to the Generalized Regression Platform and run this. And the estimation method that I'm going to use is, so when I was doing it one at a time, I was doing a lasso model, but what we can do for SVEM is use a SVEM lasso. And so that's going to say, okay, well, how many times do you want to run that? The default is 200. You can put in as many or as few as you want, and then you can click go. And so it's recalculating all those different models, and now we've summarized all 300 models into one. And so our prediction profiler is the composite model. So this is that ensemble model results. We actually have parameter estimates from every single model. So model 1 had these parameter estimates, model 2 had those. So if you really wanted to get into the weeds, you could find out what happened there. But now at the end of the day, we have a self-validated model. And in this case, we can see that we've got a main effect here of that methanol, and that propanol squared term has been highlighted as an active effect. And one way that I like to take a look at this is because this model that we've built still has all of these terms in it. So any non-zero term is in this model. And one way to evaluate this is to look at what are the most frequently occurring model terms. So I can just sort by this column. And so the intercept is in all of the models. And then methanol showed up in nearly 90% of the models, followed by this ethanol time, and then time main effect. And so we can see, and maybe we have a threshold cutoff for what model terms we would want to put in. And while this is not necessarily going to tell you what the truth is of the system, it's certainly giving you more insights. It's another tool that you can use to find out what model terms might be active, and what the variance is on their parameter estimate. And I'll just summarize with a chart here on SVEM and show what you can do. So you can take this data table and you can just take it into Graph Builder. And what I like to do is visualize my parameter estimates this way. So I've got my intercept here. So what I've done is, these are the scaled estimates. So the larger the magnitude of the estimate, the larger the effect. The error bar is the standard deviation on that. And then the color is the frequency that it occurs. So those that are red really didn't show up a whole lot in the models, and the ones that are green showed up more frequently. And so now we can see, what model effects are really showing up most frequently. And for those model effects that have standard deviations that are spanning the zero line, maybe they aren't statistically significant. So we could really use this as a way to reduce the model as well. And maybe these are the model effects that we would want to build on, either the largest magnitude positive or the largest magnitude negative. All right. So SVM is really providing another layer of insights into the system that we might miss if all we looked at was some ordinary least squares modeling. Ultimately, you'll probably get to the right answer by doing confirmation runs, confirmation trials, or augmenting the design. But now you've got some more information to help inform those decisions. And we have some customers now that are reporting they can run up to 25% or 40% fewer trials when they use SVEM compared with traditional modeling. That's a big savings when it comes to time and money. So, SVEM is not going to be appropriate for every design though. If you want to learn more about it or want to see if it fits your system, you can certainly reach out. We'd be happy to talk with you about it. And I know, so it looks like we're right at the end of the time. So I'm going to pause here. I once again covered a lot of ground. These are two topics that I think are really game changers in JUMP when it comes to DOE setup and analysis, and happy to talk through, answer any questions that you all might have.